was in those days.
People were searching for the virus. People thought there was probably a virus there, but there had been no data that would convince a reasonable skeptic that there was a virus causing this transmissible disease, scrapie, and then, of course, Creutzfeldt-Jakob disease at the time I entered the picture.
During a series of experiments over several years, Dr. Prusner and his colleagues developed techniques that revealed the infectious agent lacked a nucleic acid and couldn't be a virus.
There was no evidence for a nucleic acid. All the evidence pointed toward a protein. It allowed us then to isolate that protein, so we now could physically study the protein. No one had ever known about this protein before, the prion protein, or PRP.
Dr. Prusner coined the term prion to denote a protein infectious particle. Now we know that normal prion proteins are abundant in the body and are found on the membranes of many cells, including neurons. As with other proteins, they're produced in the endoplasmic reticulum, processed in the Golgi apparatus, and transported to the cell surface. And as with many other surface-bound proteins, they're engulfed by endocytosis and degraded within lysosomes. The normal cellular prion proteins are designated by the term PRPC. The three-dimensional structure of these proteins is predominantly made up of alpha helical configurations. In prion diseases, that configuration changes to become predominantly beta-pleated sheets. The abnormally folded prion molecule is designated PRPSC because it was first identified in scrapie-infected sheep. An abnormal prion molecule can induce other normal prion proteins to adopt the pathologic configuration, thus leading to a geometric increase in the number of abnormally folded prions. These molecules aggregate together. They're resistant to proteolytic digestion, and so they can accumulate within lysosomes or on the surfaces of the cells. They're also deposited in the extracellular space and can be stained with an immunostain using an antibody directed against the prion protein. In this section, the brown areas represent deposits of prion protein in a patient's brain.
That protein, when it accumulates, is what causes the disease. The malfunction of the nervous system is really the result of PRPSC accumulation.
One of the great paradoxes about these diseases, which is unique, is that they're sporadic, like sporadic CJD, they're transmissible, like iatrogenic CJD, and yet they're inherited as well. And that's always been a major problem to understand. And one of the attractions of the prion hypothesis is that it unifies all these different types of CJD by explaining it all due to instability in the protein.
The discovery that prion diseases can be genetic, they can be infectious, and they can be sporadic is a whole new disease paradigm that no one understood before this was teased out.
A spontaneous change in conformation sets off the process that leads to sporadic prion diseases, and those make up the vast majority of cases. In the familial forms, a mutation in the prion protein gene is responsible. The acquired or infectious forms are induced when abnormal prions from the outside gain access to the CNS. Those include iatrogenic CJD, Kuru, and variant CJD.